Hello, I'm Alex Satz, and I've been working in the DEL field for about 15 years. And in this presentation, I'm going to be talking to you about how we at WuXi do DEL. And I'm going to touch upon a couple of other different topics. I'll give you a case example of the successful screening of a target to find a drug-like hit. And I'll also talk about a couple of limitations that DEL screening currently has, and how we in the field are attempting to tackle those problems. So I'm part of a WuXi HITS team, and what we do is everything in the early part of the drug discovery world. So we produce proteins, we do screens, including DEL screens and fragment-based screens, biophysical follow-up work, and crystallography as well. So everything that's needed in order to find hits against a target, prove that the hits are real, and then enable a project team to proceed forward. Okay, so of course, a big part of finding hits are running the DEL screens, and that's the area that I've worked in for quite some time. And I'll very briefly talk about how we at WuXi do this. So a generic picture is shown here in this slide, and we see the barcode that we use. And when we sequence the barcode, that tells us what the chemical structure is, which is attached to the barcode through a long flexible tether. And so in this manner, we can have a mixture of different library members, and we can always sequence the barcode, and we can tell what the chemical structures are. So we make these DELs by way of split and pool combi-chem. So for instance, let's say we have a mixture of 3 different library members. So we have 3 different barcodes, which encode 3 different structures. And we're going to take this mix and we're going to split it up into 3 different tubes. So that's the split part of split and pool combi-chem. And then in each one of these different tubes, we're going to add a different building block, which we call a BV, and a different tag. And so the tag is ligated onto the end of the barcode. So the tag is ligated on here. We see a blue tag, and here it is. We see a red tag, and here's your red tag. And we do the chemical step on the other side. So we're building up a chemical structure at the same time that we're building up the barcode. And so we've started with a mixture of 3 different things that we split into 3 different tubes, and then a different BV and tag in each one of those tubes, to now create 9 different products. And what's nice about having a barcode is that you can then combine all of these different products into a single tube, so you're repooling them. And at any point in time, you can sequence all of the barcodes in the tube, and you know the relative amounts of each one of the chemical structures. So we do this, we make very large DELs. So our split sizes, instead of being a split size of 3, might be a split size of 3,000. And in this case, in the picture that I'm showing you here, we have a DEL kit. And in each one of these tubes, we have a mixture of 50 different DELs that we have made, for a total of 14 billion different encoded structures. And we have 4 tubes because we have an essentially identical copy of these 14 billion structures in each one of these tubes. And that allows you, who receives the kit, to then run a screen underneath 4 different conditions using these 14 billion different structures. So for instance, in how this DEL kit works is you get the kit from us, you run the screen in your lab, you send the wet samples back to us, we do the PCR, we do the informatics, and then we provide the structures of the hits back to you. Okay, so how is the DEL screen itself done? So the DEL screen is basically a binding assay. So we take the target protein and we mobilize it onto a solid phase resin. We then take the DEL from one of those tubes. So each DEL is in this tube. We take the DEL from the tube itself and we simply add it to your target. Now, most library members in that tube won't bind to the target, so those get washed away. But the ones that are bound, we collect those and we elute them. We actually take this elution and we add it to fresh protein again, and we go through this cycle 2 to 3 times. But in the end, we take the library members that were bound to your target and we sequence the barcode. And from that sequence, we determine what the structure was. All right, so I'm going to give you a case example which shows you sort of how a project team might use DEL in order to find a hit. So the example here is a published one. I give the citation here. And this was against a transmembrane receptor kinase called DDR1. So first I'll talk a little bit about what the target was and why we chose it, how the screening went, the med-chem follow-up work, and finally the in vivo result. Okay, so why DDR1? Well, so gene knockout models suggested that DDR1 would be a good target for fibrosis. So for instance, indications such as CKD. Now, the indications that the target team or the project team wanted to go after were long-term. And so they knew that they wanted a hit against DDR1, which would be selective for it relative to DDR2. However, the literature really didn't contain any known hits that were selective for DDR1 versus DDR2. And so they knew that the biggest challenge of the project would be to find something selective. So to address that challenge, the project team came up with several different ways of potentially finding hits that would be selective for DDR1. So one of those things was structure-based. There were a lot of known crystal structures of DDR1 with inhibitors bound. But of course, the problem was that these hits were also very active against DDR2. But the idea was that you could look at the crystal structure and perhaps with rational design new structures which would be selective for DDR1. The second way was to run an HTS. And then the third way was to run a DEL screen. And so to make this long story short here, the only screening method which ended up resulting in hits which were selective for DDR1 over DDR2 ended up being the DEL screen, even though many different methods were tried. And so I'll talk more about the outcome of the DEL screen. So I'm showing you here two different cubes. So we have the building block, which was used at the first cycle of chemistry during the production of the DEL, the building block, which was used at the second cycle during the production of the DEL versus the enrichment levels. And what we see here is that there are many different clusters which enriched against DDR2, but in the same screen against or they enriched against DDR1, but they did not enrich against DDR2. So in total, we had 15 different clusters showing up against DDR1 that didn't show up against DDR2. And of course, it made sense to follow up with those clusters first because they had the best chance of being selective for DDR1. Just going to show you one example of one of those clusters, what we called cluster 2. So if you look at the different building blocks in cluster 2, which were attached to this scaffold shown here, you see that structurally they all look a little bit alike. So there is some SAR there. And one of these structures, so we call this compound 2A, was indeed selective for DDR1 over DDR2 by greater than 100-fold. Now, this hit wasn't by itself terribly potent. It was single-digit micromolar potent. So weakly potent, but selective. We then quickly got a crystal structure of this DELT hit. And since we had worked with this target and its crystal structures quite a bit in the past, we knew that there was an unoccupied back pocket here. And so the very first compound that we made based upon the DELT hit was to add a CF3 group here to extend into the back pocket. So the CF3 group, this is the R group, and this is the R group shown here. And we immediately went from a hit, which was weakly potent, to one which was nanomolar potent. We also knew from previous structures that we had worked with that there was a good chance that we could convert the carbon here to something else and that we could maintain its potency. And indeed, we found that the potency was maintained, but the selectivity was now 400-fold for DDR1 over DDR2. And so at this point in time, this was one of the most potent and selective hits that the project team had ever found. And when you scan this hit against the entire kinome, what you find is that it hits DDR1, it hits DDR2 a little bit, it barely touches upon two others, and otherwise this hit is completely clean. And so it doesn't hit any other kinases. So at this point in time, we had a potent hit which was selective, but it wasn't ready for in vivo use. And its problem was mainly that its clearance was rather high. And to address this, we had to make a scaffold change, as shown here, to produce our new lead 2.54, which was just as potent, just as selective, but now the clearance problem was solved. And now we were ready to run an in vivo model. Okay, so here I'm going to show you two plots describing the in vivo results of dosing mice with compound 2.45. And so we have the healthy wild-type mouse, the plus, and we have the disease model mouse, the double knockout. And what we show here in this plot is that the total expression levels of DDR1 remain high in the knockout mouse, whether or not compound 2.45 is added. However, DDR1 autophosphorylates itself. And so when you inhibit the autophosphorylation of DDR1 with compound 2.45, we see now the levels of phosphorylated DDR1 with the addition of the compound 2.45 is reduced to the same level as that of the healthy mouse. Okay, so I've just showed you a successful example of running what we would call a normal DEL screen. So we have a project team, we have a relevant target, we find a drug-like hit, and that drug-like hit is brought forward into an in vivo model. Okay. So at this point in time, you could say, well, DEL works, DEL is perfect, you know, and there's no other problems to be solved. But that isn't quite true. So DEL does have certain limits in how it's currently done. And so because there is a covalent linkage between the ligand and the bead, or the ligand and the barcode, we can't ever get the ligand inside of a cell. So it's very hard to run a cellular functional screen using DELs as they're currently used. It's also very tricky to run a biochemical screen. And again, that's for the same exact reason, because the ligand is covalently attached to its barcode. So the way to get around that is to make these one-bead-one-compound DELs. And in this case, the ligand is attached to the bead, and a bead is attached to the barcode that encodes the ligand. And in this case, you can put the bead in a very small picoliter droplet of water, and you can photocleave the ligand from the bead. Now the bead is free to travel in solution and interact with the target or a cell. And the connection between the ligand and the barcode is kept because both are trapped within this picoliter aqueous droplet. So what we've done here is we've taken the ligand, which we found in this previous DEL screen for DDR1, and we've converted this into an FP probe. And when we take our probe and we titrate it with DDR1, we see that the FP signal goes up. Additionally, if we take our probe and we put it in droplets of water in a microfluidic chip, and those droplets travel through the chip and their FP signal is measured at the end of this chip, we can then take thousands of droplets and we can measure the FP signal in each one of those droplets, and we get a distribution that looks like this, where the average FP signal is about 0. But we can also form those same droplets with the probe and DDR1 combined. And in this case, when we look at the FP signal in each one of those droplets, we get a distribution as shown here. And now the average FP signal is much greater. We see that there's a nice clean baseline between these two distributions, and we judge that this is an assay that's good enough to run a full screen on. And so I should have mentioned that the heavy lifting of this work was done by the Brian Paco lab. He's now at UCI. And this was a collaboration between the Paco lab and myself when I was at Roche. Okay, so Brian and I, we make a small 60,000 member OBOC DEL, and we screen this DEL against DDR1, and we find the hits that you would expect. So hits such as this, I've actually shown you this exact structure which came out of our initial DEL screen. And we also found hits which we expected to find. A hit such as this is known to hit targets such as DDR1. And we also found certain hits which we hadn't ever seen in the past as well. And we synthesized some of these hits, and we found that they too were indeed active against DDR1. And so we use this as a proof of concept to show that this type of screen could work. It's one example of 2 that we have worked on and that are published. And we think that this is the first step towards what's going to be a multi-step process towards cellular functional screens. Okay, so now I want to switch gears and talk about a second limitation that we in the DEL field have. There are a lot of chemical structures that we would like to make, but because the chemistries that we use to make these structures need to be compatible with the barcode that we're using, there's a lot of chemistries that we can't use. So chemistries that require strong acids, strong oxidants, really high heat, reactions that require all of the water to be gone, those are very tough for us. However, in the last few years, there are more and more papers coming out with new ways that we can make DELs, and I'm just going to quickly show you 2 slides, 2 different examples, and I'll try to touch upon the highlights of why I think these examples are quite good. So the first case here is a collaboration between WuXi and AbbVie. So what is nice about the chemistry as shown here? Well, first, the amine building block itself is very large. So there's thousands of these that you can purchase, and so you can make a lot of different structures from them. The second thing is that the reaction conditions themselves are extremely mild. So this reaction is done at room temp. And the third thing is that the product you make from a medchem drug-like perspective, the sulfonamides are perfectly fine structures, and that's important too. So they're stable, and they won't cause the project team any problems. So another nice case is the Van Leusen imidazole synthesis. This reaction certainly is not new, but its application to DEL is. So here we're forming a very small core. So a 5-membered ring is about the smallest het-arrow ring that you can possibly form. And the diversity around this ring is quite great. So Granted, the Tosmic only has a split size of 5. The diversity there isn't that great, but the diversity at the other 2 points is quite high. And so these structures are achieving a lot of the things which we know in the DEL world are the kinds of DELs that provide drug-like hits. And that is the diversity is high, the molecular weight is low, and it's a rigid structure. And so we know that when we make structures that are really floppy, that those tend to not provide many potent hits, but more rigid structures tend to provide more potent hits. Okay, so those are just 2 cases that I thought I would outline. I would point out that we at WuXi, and in part in collaboration with the Lu Lab in Shanghai, we have published many more papers on this topic in just the last year. And so I've listed these here, and since this is a video, you can always pause and look up these different papers if you would like. Okay, so as a last point, I want to say that if you're a professor or you're with an academic group, you might have a target that you want to screen, and you might think that it's great that DEL works and that DEL can provide drug-like hits, but you might not have access to any DELs to screen. So we at WuXi, we have solved this. You can go to the delopen.org website and you can order a DEL kit for free. So we send you the kit, you do the screen in your lab, you send the samples back to us, we do all the other work, and we provide the hits to you. It's basically free. If there are any active hits that come out of the screen that you like, then you completely own those hits. And so at that point in time, you are free to get a patent, you are free to sell those hits, you are free to publish a paper with those hits. There are no strings attached. All right, so the acknowledgements for what I've talked about, I've worked with many different groups around the world, many different academic groups as well. Since most of this is published work, I simply list here all the authors from those papers. So there are quite a few. I would also like to thank the team that I work with. The WuXi DEL screening team has over 130 people working on it, which I am just one of these. And I'm showing you a few pictures of the management team for the WuXi DEL team. Okay, so thank you very much, and I look forward to meeting most of you at the later breakout sessions.